This is the beginning of the second video for the signaling chapter, and in this video we will focus on the receptor tyrosine kinase pathway. So receptor tyrosine kinases generally regulate pathways that control cell growth, and primarily they activate cell growth by promoting progression through the cell cycle in response to ligands called growth factors. So when these growth factors bind, they tend to drive cell division. Because of this fact, many of the proteins of the receptor tyrosine kinase pathway also serve as oncogenes. So when this pathway becomes hyperactivated, that can lead to cancer. So to understand the receptor tyrosine kinase pathway, we first need to understand receptor tyrosine kinases. So they're known as receptor tyrosine kinases, so they're receptors, so they have a ligand binding domain. But the tyrosine kinase part is that they have many tyrosines in their cytoplasmic domain, and the cytoplasmic domain acts as a kinase, meaning that it can phosphorylate tyrosines. So it can phosphorylate these tyrosines in the tail. So receptor tyrosine kinases can phosphorylate one another on the amino acid tyrosine. When a ligand binds this pair of receptors, so these always act in pairs, so when two ligands bind, these receptors dimerize, meaning they come together to form a dimer or a group of two receptor tyrosine kinases, and then they phosphorylate one another on tyrosines. So now this fully activated pair of receptor tyrosine kinases has phosphates on all of these tyrosines in the cytoplasmic domain, and then that leads to cellular responses. So now that we understand a little bit about receptor tyrosine kinase activation, I'd like to go through the broader pathway. So in the receptor tyrosine kinase pathway, once again, when the receptor binds the signal molecule, the growth factor in this case, the receptors then dimerize, and that leads to phosphorylation of tyrosines in the cytoplasmic domain. And then an adapter protein called GRAB2 binds the phosphorylated tyrosine, and it acts as a scaffold to recruit another protein, SOS. And SOS is a protein that activates the GTPase RAS. And you may remember we briefly talked about GTPases in an earlier chapter. So RAS is a protein that becomes active when it binds to GTP. So SOS causes RAS to release GDP and bind GTP. RAS then activates a kinase cascade. So first we have a MAP kinase. So this is a mitogen activated protein, kinase. It phosphorylates and activates MAP kinase, which phosphorylates and activates MAP kinase. MAP kinase then enters the nucleus where it works with transcription factors to control expression of specific genes. So ultimately, the response in this pathway is a change in gene expression. So I want to highlight a few important pieces of this pathway. So just to review, when the receptor becomes activated, the two receptors dimerize and phosphorylate one another, so they autophosphorylate one another on tyrosines. That leads to the binding of the adapter protein GRAB2. GRAB2 recruits SOS, which activates RAS by causing it to bind to GTP. RAS then activates the major source of amplification in this pathway, which is a MAP kinase cascade. And then finally, the last MAP kinase enters the nucleus where it works with and activates other transcription factors to promote changes in gene expression that drive cell division.